you can learn about them by going through various chapters. And it was difficult for me to kind of make a reading assignment based off of that. But I think you learn a lot by just reading the viruses chapter, because now with everything that you do know about the drugs, about the life cycle, about genetics, you know, a lot of things begin to make sense. But when it comes to the exam, I'm not going to test you on things that we didn't talk about. Okay, so don't worry about that. I will post the recordings, but I must warn you that the recordings would be unavailable, you know, before the exam. So make sure that you have watched them. You know, they will not be left up for the day of the exam. Okay. I will post today's recording, and I will, let me remind me if I forget to hit the record button. Okay. Can everybody see the slides? Okay. Yeah, okay, awesome. All right, so let's hit record.
Recording in progress.
So as I said, we'll be talking about some viruses today. I have been saying it throughout the semester that we will come back to this topic at the end of the semester, because a lot of trying to understand how viruses evolve depends on our understanding of genetics, because essentially that's what they are. They're genetic parasites that come in, bring in their genome, and then they take over the cell's machinery. So we're going to talk about different viruses today, and, you know, sort of hopefully understand how each one has its own genome and its own host. We've talked about tissue tropism, we've talked about host range, you know, and so I don't have the time to talk in a lot of detail. So hopefully you guys can go on to take the virology course, and, you know, Dr. Vesinen spends an entire lecture talking about individual viruses. So he'll talk about, you know, HIV individually, he'll talk about flu individually, he'll talk about coronaviruses individually. So a really great course if you're interested in not just the life cycle, but also the immune response and you know, how widespread something might be and things like that. So it's a really great course. Yes, virology. There's virology that's offered every, I believe every year, or maybe every 3 semesters. But, you know, it is offered, and it's a great course. So certainly take that if you're interested in learning more about the viruses. So first virus that we're going to talk about is the bacteriophage lambda. So bacteriophage lambda is a temperate virus, right? And it infects E. coli. So E. coli is its host, and E. coli lives in our gut, right? Our colon is where it's naturally found. This is its natural habitat. We are the reservoir for this particular bacterium. And this bacteriophage infects E. coli, right? So it's also found within us. And we know how temperate phages life cycle works, right? We know that it has these, you know, 2 life style choices. It can either infect its host cell, which is going to be E. coli. We're not the host cell for the phage, so we don't have to worry about it. The phage is a DNA virus, and so it can either enter the lytic cycle after it has infected its host cell. And we know that phages, because of their complex capsid, as well as the complexity of the cell envelope of the bacteria, only inject their DNA into the bacterium, right? So in this case, we're just seeing the DNA being injected. And once the DNA is within the cell, it directs the cellular machinery to synthesize, you know, new virions, and then eventually these cell wall weakens due to production of viral enzymes, and then these cells will lyse, and the new virions can go on and infect brand new cells. So this is our lytic cycle of a bacteriophage. And then you can have the same phage enter the lysogenic cycle, where once the DNA is injected, instead of it controlling these host cell into producing new virions, it gets incorporated into the host genome in what we refer to as a prophage state. And we refer to the cell as the lysogen, I hope you remember. And there were several advantages, but one of the main advantages was that, every time the cell would undergo duplication, the phage will be duplicated as well. And of course, if it's a cell that is metabolically inactive, right, so that it doesn't have a lot of transcription and translation going on, then it makes sense for this prophage to kind of, hide and kind of stick around until cell becomes active, right? And we talked about the process of induction, that if the cell somehow was on path to dying, right? UV exposure being one of such stresses that can be detected on a, as a trigger for death. And so that is a signal for this prophage DNA to excise itself out and then quickly undergo the lytic cycle, right? So that it's a survival strategy for the virus. The bacterium is about to die. If I remain as a prophage, right, there's no further infectious cycle for this virus. This virus needs to be in its virion form. It needs to have all those proteins, as part of its complex capsid so that it could infect the subsequent cell. So, it has to sort of make these decisions, and it's a very complex life cycle for the virus. And like I said, I don't have time to talk about all the details. And there is obviously a lot of signals that are being sensed from the environment, from the host cell as well. We don't necessarily understand how viruses are able to sense that, but obviously for a virus to go from a prophage to undergo induction and excise itself out, it has to be detecting something, right? It has to know that the cell is under stress and it's about to die. So there's a lot that we don't necessarily know. And, so there's a lot that still needs to be learned. But let's today, look at its genome a little bit. And then, how it decides between the lytic and the lysogenic cycle. Again, there are a lot of genes and we don't have the time to talk about all of that. So we're going to talk about just a regulatory mechanism that allows this virus to switch from lytic to the lysogenic cycle. So bacteriophage lambda, if you've ever worked in a laboratory and you have used lambda DNA standards, which are used as a marker to determine how far the DNA has traveled on a agarose gel. It's essentially just Hindi 3-treated lambda bacteriophage DNA, which gives you these different size DNA fragments. And then we know exactly what size they are, and we can use them as a standard to measure the size of unknown pieces of DNA fragments. So we use it if you've ever taken any of those DNA courses, like the lab courses, 362, or if you've worked in a laboratory, you're aware that this is used often in genetic engineering labs, wherever biotechnology is involved. So it has a pretty large genome, and it's interesting in that it can either exist as a linear piece, as you could see at the top, or it could become circular and it can exist as a circular piece. So generally within the virions, as it's being packaged into the heads, it would be found in this linear form. But during the lytic cycle, originally when the copying of this DNA begins, you'd remember that bacterial genome is circular, and we talked about how there is this end replication problem, and so you want to avoid that end replication problem. And so the DNA is converted from being circular, from linear into circular, so that the E. coli can easily copy it. And so when we look at the ends of this linear piece of DNA, there are sites that we refer to as cause sites, which stands for cohesive sites. So they are, inverted repeats where they can complementary base pair with each other. And so that is what converts this linear piece into a circular piece. But this happens not while the DNA is within the virion, but rather while this piece of DNA is within the cell, so that the replication process could become easier. So if this piece of DNA, is going to be replicated by the viral, by the bacterial machinery, then it has to come with some sort of an origin of replication that needs to be recognized. So we have initially a early gene product. So when viruses begin to express their genome within the bacteria, you'll have, different genes that are expressed at different times. So you'll hear words like, early genes or early proteins, and then the late genes or late proteins. The early proteins often happen to be, proteins that are expressed by early genes that are involved in the earlier steps of the viral replication cycle. So they might involve things that help with the transcription or replication process for the viral genome. Okay, and when we talk about the late, genes, these are going to be genes which are expressed to, finally package and fully synthesize the virions. So all the capsid genes and the tail genes and things like that, they will be expressed, towards the end of the virus replication cycle or life cycle. So it, depending upon when a protein is needed, generally those that are involved in transcription or replication or controlling the host machinery will be expressed very early on. So we call them early genes, and those that are involved in assembly of virions and their release from the host cell, those will be needed, towards the end of the life cycle of the virus. So we refer to them as the late genes. So here we have a gene O product. So this is obviously, an early gene, something that is expressed very early on, and that's going to help initiate the process of replication. And when you look at the replication process, it's a bidirectional replication, something very similar to what we've seen in bacteria, right? So it's going to form that data structure that we talked about. And once the two replication forks run into each other, it's gonna sort of the replication process ends, but you still have to kind of detangle the two circular genomes that are joined together. So we've talked about topoisomerase as being involved in that process as well. So, even if things are not mentioned here, I hope you understand that because the host cell is a bacterium, it's limited by, its own enzymes in terms of what it is able to do or what it isn't able to do. Okay, so it is the phage is using the bacterial host machinery. So, a lot of those same steps are going to be involved. The same proteins are involved. The same enzymes are involved. Okay, but once you have sort of these two genomic DNA resolved from being tangled, the rolling circle method of replication begins. And we've talked about it, that you're going to have an enzyme that comes in to clip, right, one of the strands, and it's generally the outer strand, and the hydroxyl on the 3' end of this nicked DNA is going to act as the primer, which will then be sort of extended, and this extension causes the outer strand to kind of displace itself, okay, as it grows around the inner strand as a template. So inner circular strand is being used as a template, okay, the inner strand is being used as a template. The outer strand is going to use its 3' hydroxyl end to continue to sort of grow. And then as it's doing that, it's going to continue to replace, whatever was there before. And so This process we call the rolling circle method of replication. Hopefully you remember that. And so this is going to continue to replicate the viral DNA, okay? And so you're going to have not a single unit production of DNA, but you're going to have multiple units of phage DNA that are connected together. And so we refer to these as concatemers. And eventually these concatemers will also be converted into a double strand. And you're going to have, as these concatemers are being packaged into the virion heads towards the end of the life cycle of the virus, you're going to have the terminase complex that's going to determine exactly where those cohesive sites are, and it's going to make that staggered cut so that you restore those cohesive sites that you had at the beginning of this life cycle. So although you have several genomic units that are being synthesized together, the terminase enzyme complex is going to ensure that only a single unit is packaged into an individual virion head, which of course we've talked over and over about that. That's such a compact space that it can really just fit that one unit of the genome. So it's not going to fit more than one unit. If you try to fit in two, you're just not going to be able to synthesize functional virions that are going to go on to be able to infect new host cells, because their synthesis might not even be completed. So this is essentially the replication of the genomic DNA. Remember, replication also refers to the entire life cycle. So if you hear me use it interchangeably, don't get confused. So this is how the genome would be copied. Now, when we look at the genome of the bacteriophage, there's lots of these genes here, right? And I'm not asking that you memorize all of them. Like I said initially, we're going to just talk about the regulatory mechanism that allows the phage to go between the two lifestyles. But I want you to notice that there are some genes here that are colored in lighter tan, and then there are others that are colored in orange. And so those that are colored in orange are generally the ones that are involved in sort of the regulation of whether you enter the lytic stage or the lysogenic state. All right, so sort of that regulatory decision making. And when we talk about a lot of these tan genes, notice that they're involved in synthesis of head proteins, synthesis of the tail proteins, lysis proteins, right? Excisionase that's going to help excise out the prophage DNA from the bacterial genome. So, they're sort of clustered together, and, it makes sense because, they will be synthesized together. And often they might even be sharing a single promoter, so that they might have a single transcript that is being made, which then can be translated into multiple different proteins. Okay, so there's lots of these genes here. This ATT site, by the way, is the site that is used by this phage to insert itself into bacterial genome via recombinant homologous recombination. Okay, so that's that. Now, a few genes here, although not so visible here, they have overlapping coding regions, and so that doesn't necessarily mean that the genes are present on the same strand of the DNA. You could have maybe one of the genes on the top strand and the going in one direction, and the coding region for the second gene on the bottom strand going in the opposite direction, or they might even be going in the same direction, but they just overlap in a small region, right? So that's a common feature we see in viruses a lot, that their genomes are so compact. Not so much the case for phage lambda, because it has a genome that is 48.5 kb, which refers to kilobases, and each base is a nucleotide essentially. So it's a rather large genome, and so there isn't always a lot of overlap, but things like HIV, they're going to have a lot of overlap because their genome is extremely small. And so just sort of we're going to talk about basically this area here. Okay, you know what's found external to that. You're going to have genes that are involved in synthesizing proteins that allow you to finally assemble the viruses. And since we're worried about the decision making, we're really going to be focused on these orange genes. And notice they have their own operators, they have their promoters. There's a PR, it means that you have, it's going to be the direction of transcription is towards the right. You have the PL, which means that the direction of the transcription is towards the left of it. So, and then you have PRE, which means that it's an earlier promoter going right. Okay, so, there's a lot of terminology here. There's a lot of genes here, not asking that you memorize all of them, but at least know the ones that we're going to talk about. So this region essentially just shows you basically this area just zoomed in. Okay, and so we're going to look at, how lysogeny or lytic cycle is determined. And so notice you have the CRO gene, the C2 gene, the C1, the C3 genes, the excisionase, the integrase gene, and they have their own sort of promoters. You have the PR and the PRE, so the transcription is going to be towards the right, and PL, the transcription is going to be towards the left. And then you have these T's, TLs, and TR. And so these refer to terminators. These can be, a terminator if you start transcribing from this promoter, this transcription can end at this first terminator site, starting from PL to TL. All right, so that would be the length of the transcript. And then you again have several terminators on the other side. So if you started with PR, you can have a termination at the TR site, or you have the transcription start from PRE, and you can have termination occur at the TR2. So it's showing you a lot of that. And then the operators, which obviously bind some sort of a transcription factor, right? So it could be either an activator protein or a repressor protein. But notice how this is a pretty congested area. There's a lot of overlap, right? So there's lots of genes, there's lots of promoters, and lots of terminators, a lot going on there. Now, initially, when virus injects its DNA into the bacterium, one of the first things that begin to be transcribed are the promoters associated with PR and PL, okay? And so when these are being transcribed, the only sort of thing that is being fully synthesized is this N protein and the CRO protein. Okay, so those are the two that are being synthesized earlier on. So these are your, early gene products. And one of the things with starting transcription at PR and PL is that you have these terminators to worry about so that the transcription can end there. And so to prevent transcription from ending there and extending beyond that, right, so that PL can go and transcribe the DNA corresponding to gene N and the C3, right, means that transcription has to go beyond this first terminator, right? So the transcription has to extend. And in order for that to happen, you need to have you need to synthesize the N protein, and the N protein has to act as the anti-terminator. So it prevents termination at the TR and the TL sites. Okay, so I hope that makes sense. Okay, so that initially you start off transcription from promoters that can, end very early, sort of prematurely. And in order to prevent that premature termination of transcription and to be able to, synthesize the transcript, which will lead to synthesis of proteins like C3, for example, you need to have this initial transcript be translated into the N protein. And that N protein would then act as an anti-terminator, preventing the transcription machinery from ending transcription at the TL site and the TR site. Okay, hopefully that makes sense. Questions on that? Okay, all right, so once you have this transcription that is able to extend beyond it, now you're able to synthesize, other proteins. You're able to synthesize C3 protein, you're able to synthesize a C2 protein, right? As well as CRO protein, the N protein is there and so on. So, we're gonna see now that there is a competition between a couple of proteins that determines which lifestyle would be adopted. Would it be the lysogenic or would it be the lytic cycle? So it all has to do with the accumulation of a particular protein C2. All right, so C2 is right here. So you would initially have CRO, and once you have the anti-termination happen, then you will also begin to synthesize C2 protein and C3 protein. Let's say earlier on in the life cycle, the C2 protein accumulates. Now, think about C2 protein before I go forward, is that it's labile. It's degraded by the E. coli enzyme. It's a protease called HFLB. Okay, so the C2 protein has to somehow be protected by the host's protease. All right, and if a cell can do that, then you can have one lifestyle. If the cell cannot protect the C2 protein, then it will go for the second lifestyle. So initially, let's say that you have high levels of C2 protein and high levels of C3 protein accumulate. So there's some sort of, like I mentioned, there's a need to protect C2 from the host protease, and the viral protein that protects C2 is the C3 protein. All right, so C3 protein is being synthesized from the left promoter, and C2 protein is being synthesized from the right promoter. And so once you have lots of this C2 and C3 protein accumulate, then it's no longer labile. C2 is no longer labile. It's now protected from the host protease, and it can act together with C3 to turn on transcription from other promoters. So we've already seen a transcription from PR and PL. Now transcription is going to start from other promoters like PRE and PI. And if you look at the PI, it's basically going to result in synthesis of the integrase protein. Okay. And when we look at the PRE, it's going to result in synthesis of the C1 protein. So C2 and C3 combine together, right, and activate transcription of genes that eventually code for the integrase protein and the C1 protein. So the role of integrase is clear, right? It integrates the viral DNA into the bacterial genome. But what's the role of the C1? All right, so C1 is referred to as the lambda repressor, and it basically represses transcription of all other viral promoters, right? So it stops transcription from all other viral promoters, so the PR and the PL that we've looked at that eventually lead to synthesis of C2 and C3 and CRO and all of that. So that's all shut down. So it acts as a repressor for all of them. But it also, acts as an activator when it comes to its own promoter. All right, so it acts as its own promoter, and it acts, it activates its own transcription. So it's an activator for its own promoter, for its own gene, but it acts as a repressor for every other gene there is, okay? So the end result is that once the viral DNA is inserted into the host DNA using the integrase that was synthesized, beyond that, no other viral protein other than C1, which is lambda repressor, would be made. It will turn off transcription of all other viral genes, only continue to transcribe itself and translate, of course, allow bacterial host cell to translate itself. And so you're going to have the steady population of the lambda repressor to ensure that state where no head proteins are being synthesized, no tail proteins are being synthesized, no regulatory proteins are synthesized except for itself, right? So this sounds like an inactive virus kind of hiding within the bacterial genome, okay? So, it started with initially making sure that we protect C2 protein, okay? And C3 is not labile, C2 is labile, which means that it can degrade easily. C3 and C2. So C2, if we go back, right? If we go back, C2 is being synthesized from the right promoter and C3 is being synthesized from the left promoter, okay? Now, so how does the cell initially, okay, awesome, all right. So how does these, the C2 is initially protected? So it turns out that viruses have a way of detecting the metabolic activity of the bacterium. Okay, we don't know how they do it, but they know whether a bacterium is metabolically active or not. One way to determine that is high multiplicity of infection. And you remember, it's a complex term that basically just says that there is a lot more viruses than there are host cells. Okay, and so if you have a lot of viruses and few host cells and you infect and undergo lytic cycle, then all the host cells will die and there is, no more chance to produce new virions. So what should you do? You should just lie dormant as a prophage instead, right? So that's one thing that they determine. And the other thing, of course, if a bacterial cell is metabolically slow or inactive, then that means that it has access to lower nutrients. So that could be a stage because these bacteria are found within our gut. This could be like, the time between meals, okay? Or if you are fasting, right? Or, while you're sleeping, right? So in what we loosely refer to as the starvation stage for our body as well, right? So that's the low nutrient stage for us. And that's when you're going to have the C3 come in and protect C2 by inactivating the host protease. Okay, and so it this regulatory mechanism which involves all of these proteins. Somehow, virus is able to synchronize the activity of these proteins with what's going on with the bacterial host cell, which is based off of what's going on within our body. If we're hungry, the bacteria are going to be, metabolically inactive or slow at least. They don't have as many nutrients around, and the prophage is going to lie dormant and take on the lysogeny, lifestyle. And so the important proteins, again, C2, C3, and then, of course, the lambda repressor and integrase is required for the insertion of the phage into the bacterial genome. Moving on to next slide. Now, how does the cell, let's say cells are metabolically active. Let's say the human host is actually just had a meal, and so there's lots of nutrients, and bacteria have had the chance to grow utilizing those nutrients. So the virus is going to detect a low multiplicity of infection. Again, complex term just means lots more hosts versus the viruses. And somehow it detects that there are high nutrients and there is high amount of HFLB protease present as well. And so if that's the case, C2 protein is degraded very, very easily because you have lots of protease around. Okay, it could just be, I'm not sure of this, but it makes sense that if a bacterial cell is metabolically slow, it's not actively transcribing and translating that particular protease. I'm not sure if the protease is degraded very, very quickly, or if it's also regulated where it's, goes between on and off switch, depending upon the nutritional status of the bacterium. But either way, you have a lot more active host protease when there is lots of nutrients, there are lots of bacteria around. And in that case, C2 protein would be very easily degraded. Okay, so C protein is very easily degraded at the very beginning of the viral replication. And instead of C2 building up, the CRO protein begins to build up. Okay, the CRO protein begins to build up. And the CRO protein is interesting, is that it's also both an activator and a repressor. So it's gonna, what CRO protein does is that it's gonna compete with the C2 protein. If you have a lot of it, then it's going to repress the transcription of genes that are involved in the lysogenic cycle, which would be your C2, C3, and the integrase things that we just talked about, and C1, of course. And it promotes, of course, its own transcription. So there's lots of CRO that is being made. You have the anti-terminator that still is being made, but in this case, the anti-terminator is turning on transcription of genes that are, towards the right of the PR. So PRE, for example, that promoter is being turned on, PAQ is being turned on, PR prime is being turned on. So genes that are towards the right, and you'd remember that those are genes that are involved in synthesis of head protein and things like that. So you have, other genes that are being synthesized or transcribed. C2, you could argue, well, C2 is still being synthesized, but it's no longer a problem because it can't be protected by C3. C3, right, is no longer being synthesized because CRO is acting as a repressor for PL. So none of these genes are being, transcribed anymore and translated anymore. So the result is that you're going to have the transcription of now genes that are involved in the lytic cycle, right? CRO is going to inhibit transcription of genes, specifically C3 and the lambda repressor, that will help to maintain the lysogenic cycle, right? If you don't have the lambda repressor, then you can't repress the transcription of all the genes that are involved in the lytic cycle. So CRO and lambda repressor, CRO and C1, work in opposition to one another. Both are activators and repressors. They act as activators for their own genes, but repressor of either the lytic or the lysogenic genes, depending upon where their role really lies. So in this case, CRO's role is to repress transcription of lysogenic genes. And so it also increases transcription of an important gene called the Q gene, okay? And so Q is an activator that's going to go on to induce transcription of late genes, the head proteins and the tail proteins and things like that. So initially, when the virus infects the bacterial cell, it can make these decisions based off of the nutritional status of the cell, the amount of protease that is present, and it either stabilizes its C2 protein or doesn't stabilize it, right? And if it stabilizes its C2 and it builds up, then you have lysogenic cycle. If the C2 doesn't stabilize and it's degraded, then CRO builds up, and CRO leads to the lytic cycle. Okay. Now, what happens when the cell finally decides to virus decides to undergo induction, so that it wants to reverse lysogeny, it wants to excite its DNA outside of the genome and undergo, the lytic cycle. And so originally, when we talk about the lysogeny, the lambda repressor has to be bound. And so now the viral DNA is part of bacterial DNA. And so whatever is going on within the bacterial DNA will be going on within the viral DNA as well. So the lambda repressor is bound to all of these promoters that correspond to the viral DNA, like the PL and the PR and things like that. And when we talk about induction, one of the things that we mentioned was UV light. The UV light is a trigger for induction, so that it allows the virus to somehow detect that bacterium is under stress, and then it begins to undergo the excision process, and then start the lytic cycle. And so one of the things that we've talked about already is that when bacteria are exposed to UV light, they begin to bring together a lot of repair mechanisms. And especially if you expose bacteria for, longer periods of times to UV light, then they bring in their SOS response. And as part of the SOS response, we talked about the REC-A protein, which kind of glides along the DNA and kind of, cleaves off any of the repressors that might be bound there, allowing transcription to quickly, begin, and then the corresponding proteins are synthesized, and then hopefully, those proteins would help with the survival of the cell. And so as the REC-A is sort of gliding around the bacterial DNA, it's going to encounter the viral DNA as well, because it's part of the bacterial DNA at the moment. And it's going to encounter the lambda repressor, and it's going to cleave it as well, so that it's no longer there. And when you don't have the lambda repressor there, then the C1 transcription, because the repressor protein was an activator for its own gene. So the C1 transcription, which is the lambda repressor transcription, is going to go down, and you're going to have lower levels of lambda repressor. So once you cleave it, you don't have additional lambda repressor to replace it, because the transcription has been shut down. Okay. And so now, because the repressor isn't there anymore, transcription can begin from, other promoters on the viral genome as well. And one of the genes, important genes that is, of course, at the very beginning synthesized is the not gene, rather the protein corresponding to the gene is synthesized is the excisionase gene. And so it's transcribed, its protein is translated, the protein is going to help cut out the viral genome at those COS sites, and then that linear piece is going to be circularized, and then the cell is going to perform the rolling circle method of replication of that, and then you would have the filling of the heads and so on. So you have the excisionase gene just kind of reverse that integration that was done by the integrase, and the at the same time, because the C2 is not there, you're going to have the protein levels that begin to increase. The lambda repressor would be blocked, and the lytic cycle would proceed with things that we have already mentioned. So, questions on this? Okay, so this is sort of an overview of what's going on, the same thing, just a little bit simplified, just the entire life cycle. So you go from the phage binding to the bacterial receptors, and these receptors happen to be maltose porin proteins. And so it binds a porin protein. E. coli is a gram-negative bacterium, so it's binding to the porin proteins that are found in the outer membrane of this gram-negative bacterium. DNA is injected, it will undergo circularization, and it will undergo transcription from the PL and the PR genes, and you can either have stabilization of the C2 by its interaction with C3, so that you have a lot of C2 compared to CRO protein. And if that's the case, then you're going to eventually have synthesis of C1 which is the lambda repressor. You're going to have synthesis of the integrase protein. It's going to help integrate the viral genome into bacterial genome, and you're going to have lysogeny. And so one of the advantages of lysogeny is that as long as you have a prophage inserted into the bacterial genome, no superinfection would be allowed. So no more viral attacks. Okay, so it's sort of a way of virus to make sure that I'm here already. This is my territory. No more viruses are allowed to come in and undergo lytic cycle while I'm waiting for things to stabilize or become ideal for this particular virus. The other condition is that you're going to have CRO in higher concentration compared to C2, and that's going to result in transcription of genes that are involved in the synthesis of the capsid proteins and such. And so you're going to have the rolling circle method of copying the genomic DNA. You're going to have synthesis of all these phage parts, and you're going to eventually have the filling in of the head via the terminase complex, which is going to ensure that only a single unit of viral DNA is packaged into individual heads, and then the cell would lyse. And often one of the proteins that the virus genome would code is what we refer to as a Holin protein or endolysine protein. So in this case, the phage has genes for two different proteins that will help to weaken the cell envelope and induce holes within the membrane, allowing the virus to leave. And eventually, of course, they can cause the lysis of the cell as well, because these holes are just good enough for the entry of the fluids from the environment as well. So a lot of osmotic pressure would eventually result in lysis of the cell as well. So you have proteins at the end helping to release these viruses into the environment as well. So people often think about this sort of regulatory mechanism that determines the lifestyle of this virus and wonder why go through such a complex life cycle. And it seems as though this viral attack, when the virus undergoes lytic cycle, is really a means of controlling the number of bacteria that you have within your gut, right? So these phages are parasitic to the bacteria for which we are the reservoir, meaning we're their house. This is where they live. This is where they multiply. And of course, we lose bacteria all the time as we defecate, but their numbers can really increase exponentially, especially after a meal. So after a meal, the prophage really enters a lytic cycle, destroying a lot of these bacteria, preventing their numbers from building up too high to the point where our immune system is not able to, basically protect us from these symbionts. And so it's a pretty interesting sort of lifestyle, and to sort of superimpose the bacterial lifestyle and human lifestyle on top of that, and really think about how these phages are actually advantageous to us is amazing. Okay, questions on any of this before we move on to the next virus? Okay, so this was a little bit more complex because there was a lot going on. With the flu virus HIV, these viruses don't have very complex lifestyles. They don't, especially flu and coronavirus, they give rise to acute syndromes, right? So they infect the individuals very quickly and cause a disease very, very quickly, right? Respiratory syndromes in both cases. And so we don't have to worry about them hiding within our system for too long and then just becoming reactivated at some point, or at least not as routinely as as E. coli would have to worry about, lambda phage. So when we look at the flu virus causes hundreds of thousands of deaths yearly around the world. And it's endemic in our population, which means that it coexists with us. Majority of us have strong immune systems, and our immune system recognizes the normally floating flu virus strains, and it learns to, the virus learns to sort of habitat with our immune system. So it basically goes by unnoticed for most people and might cause mild symptoms, but our immune system is able to help us recover from that infection rather quickly. But it does cause every now and then pandemics, right? So, the largest pandemic we know of is that of 1918, but we've had smaller pandemics since then. Now, when we look at the structure of the flu virus is interesting. A couple of things you can tell right away is that it's an enveloped virus, right? So it's going to leave the host cell via budding, right? Because it has to acquire that envelope from the host cell as it leaves, and so it's going to leave via budding, and it has all of these proteins, enveloped proteins that are embedded within it. You have the neuraminidase. We're going to talk about its function. It has the HA protein, hemagglutinin, which derives its name from the fact that it can agglutinate cells together, get them to be sticky. And you have the M2 ion channel that we're going to talk about in a moment. Here you have a drug mentioned Tamiflu, which is over the counter if you've ever taken it. It controls, basically blocks the activity of neuraminidase. So we're going to talk about what that does in particular in a minute. But so you have a lot of detail, a lot going on the outside as well. And all of that is important because that's where the receptors to get inside our cells are located. And, three of the major proteins that we'll talk about are the HA protein, the NA protein, and the M2 ion channel, which is a proton channel. And what channels do? They allow ions to go across. In this case, it's a proton channel, so it's going to allow protons to go across. And so let's turn our attention to the inside of the cell. There's a lot going on within the not the cell, rather the inside of the virus. So the core of the virus has within it a negative strand of the RNA genome. All right, so its genome is comprised of RNA, and it's segmented, meaning its genome isn't one single strand of RNA, but rather genes are located on several individual strands of RNA, and together it makes up the complete genome of the influenza virus. All right, and so it's an enveloped virus with a RNA genome that is segmented, and it's negative strand. And you'd remember the difference between negative strand and positive strand was that negative strand RNA isn't ready to be translated within the host cell. And the positive strand is ready to be translated within the host cell. So a negative strand RNA genome first needs to be converted into a positive strand RNA genome before it could be translated by the host ribosomes. Okay. In addition to that, there's a bunch of proteins. There's this matrix sort of protein that lies just under the envelope, and don't confuse it with the capsid protein. It's not a capsid protein. The RNA genome is going to be associated with capsid proteins, which are these pink proteins. RNA is represented as blue, and so we refer to individual RNA strands as a single nucleocapsid. Right, so you have a single nucleocapsid, and you have 8 of these, and then there is additional, proteins as well. We'll look at that RNA genome in this slide. So you have the 8 strands of that segmented genome. Each one of those RNA is actually folded back on itself, and it's sort of base pairing in certain regions, and it's associated with the proteins, the capsid proteins, or what we refer to as the NP proteins, or the nucleocapsid proteins. And on one end, where the two loose ends of the viral RNA genome are tied, there are these three proteins, PB1, PB2, and PA. And together, the three of them code for RNA-dependent RNA polymerase, right? And this is extremely important for the survival of the virus. Virus is bringing in to a host cell, which is our cells, RNA as genome, which needs to be converted into another RNA. Our cells don't do that. Our cells convert the information from DNA into RNA, and from there into protein. So we're asking, this virus is asking our cells to convert its RNA into plus RNA, and then that plus RNA back into the negative, or I should be calling it negative and positive, but so it started with negative, which has to be first converted into positive RNA, and then that positive RNA has to convert it back into negative so it can serve as the genome of the newly synthesized virions. Now, this is something that our cells don't do. So, and if virus was going to complete its replication cycle, then it better bring with it an enzyme that allows host cell to do that. And that is the job of the RNA-dependent RNA polymerase. And each one of these strands, notice, have with them that RNA-dependent RNA polymerase, okay? So now when we look at the individual 8 strands of viral DNA, RNA rather, viral genome, the first strand here is encoding PB1, and then you have the second strand that's coding PA, and the third strand that's coding PB2. So there are 3 separate strands, 3 separate RNA segments that are coding for that RNA-dependent RNA polymerase. And this protein was synthesized starting from these 3 genes by the previous host, right? And it was complexed with each one of these RNA segments and packaged into the new virion. Then you have the 4th segment that codes for the HA spike protein. Then you have the 5th segment that codes for the nucleocapsid, the NP proteins that will stabilize the individual RNA segment, and so on. You have the 7th that codes for the matrix protein M1. I'm sorry, the 6th. Yeah, we talked about 6. So 7th that codes for M1 as well as M2 channel. All right, so 7th has 2 open reading frames in it. 2 proteins are encoded by that segment. And same thing is true for the 8th. Okay, so that it's also going to be coding for 2 separate proteins, the NS1 and NS2, which are synthesized after the positive strand RNA is spliced. Okay, so it kind of walks you through the 8 segments that code for the 10 proteins that the entire virus is synthesized of. It must have with it as it leaves the host cell, and everything else it needs in terms of replication and of itself and translation machinery, the host will provide, our cells would provide. Now let's look at its receptor. We know that we've heard of things like flu virus, and avian flu, and we've heard of the swine flu, right? And so avian flu virus generally infects birds, and the swine flu virus generally infects the swine. And then we have also the human flu viruses. And so when we look at their structure, it's very similar to what we have talked about. So what sets them apart is the receptor. So HA spike is going to be looking for a receptor on the host cell, and that receptor happens to be a glycoprotein, so a protein that is sort of emerging from the respiratory epithelia of our respiratory tissue, and it's not just the protein that's important, but also the sugars that are attached to it. That is what glycoprotein is. It's a protein on the surface that has carbohydrates attached to it. And so generally, the receptor has a terminal sialic acid receptor or sugar. And so when we look at the flu receptors in birds, and we look at flu receptors in humans, and we look at those in the swine, what we see is that Birds have one type of receptors, so where they have the same protein receptor, they have the galactose and the sialic acid that are the terminal sugars on this protein receptor, and they're bound to each other via a particular linkage, the alpha-2,3 glycosidic linkage. And then when we look at the human receptors, the same, sort of receptor, the only difference is that we have sialic acid that is attached to galactose via a different glycosidic linkage. So it's alpha-2,6 glycosidic linkage. And that difference, although, the sugars are the same, that the way the two are attached to each other, right? They're isomers essentially. It's going to prevent certain types of flu viruses from binding to this particular receptor. So there are flu viruses like flu A, B, C that can only infect humans, whereas there are others that can only infect the birds, and then there are some that can infect both birds as well as humans. Now, swine happen to have both types of receptors in their upper respiratory tract. Okay, so they have both types of receptors. So both bird flu virus can infect them, and the human flu viruses can infect them. Okay, so we refer to swine as the mixing bowl for various viruses. Okay, why do we call it a mixing bowl? We'll come back to it in a moment. Let's just quickly look at the function of some of the viral proteins that we have mentioned already. So you're going to have the virus come in and interact with that glycoprotein with the sialic acid, right? So that's the receptor on the host cells. And that is, that receptor is going to determine the tissue tropism for this virus. Not all cells within a host will be infected. Only the ones with that particular glycoprotein are going to be infected within a host cell. So our skin is not infected with flu virus because our skin doesn't have those receptors on it. Okay, and similarly within host range as well, all the organisms that don't have that receptor, they will not be infected, and anybody who does will be infected. Right, so you have that receptor acting as the agent to which viruses would be attracted. But before viruses can bind to that receptor, the HA spike needs to undergo some modification. So our cells have a protease that interacts with the HA protein, and it cleaves it, it modifies it so that it is now able to interact with the receptor. So not just the receptor is needed for this binding, so is the protease. So if host protease isn't there and the HA can't be modified, then this attachment cannot occur. Right, so both are required. And so in any event, so once you have this receptor spike complex form, then you're going to have the receptor-mediated endocytosis, and we know what happens to that endocytic vesicle. It fuses with the lysosome to make an endosome. So that's what you're seeing. You're seeing that endocytic vesicle, that have already fused to a lysosome. And what does lysosome do? It comes with it. It brings with it a lot of acidity. So that's what you're seeing in terms of low pH and protons. And so those protons are free to diffuse through the M2 channel. Okay, and this diffusion is required because that is what leads to uncoating of the virus. Okay, and so it causes the fusion of the viral envelope with the membrane of the endosome, and so that the viral envelope becomes continuous with the endosome's membrane. They become one, and so the inside of the core of the virus now can be sort of continuous with the cytosol, and that's exactly what happens. And so that's why you're seeing these RNA genomic segments being released into the cytosol, which is essentially the uncoating process. Now, when you look at the HA during this process, the protease activated the HA so that it was able to bind to the sialic acid receptor. And HA is actually a trimer, right? So it has to sort of form this trimer. And once you have the acidity of the core increase, right, or just the acidity in general just sort of increase here, that's going to again bring a a change to the three-dimensional structure of the HA protein. It's going to, initially you had these circular globular structures. You no longer are seeing them in contact with the cell surface receptors. They've kind of, moved out of the way. And so that is essentially a conformational change for HA protein. And, there is going to be a much stronger affinity between the HA and the sialic acid receptors. The two are going to sort of be attracted, and they're going to bring together the membrane that is the host membrane, right? And the membrane that is the viral membrane. And that is what results in eventually the fusion of the two. So if you could somehow control the M2 channel, right? If you could somehow control the acidification step so that the HA doesn't undergo that conformational change, then you can prevent the fusion of the viral envelope with that of the vesicle membrane. And if that doesn't happen, then you don't see this uncoating where the viral genome would be ejected into the cytosol. So there are drugs that were used to control the M2 channel. So amantadine, which we talked about when we talked about viral drugs, is going to basically control the action of the M2 channel. If acidification doesn't occur, then fusion doesn't occur, and uncoating doesn't occur, and the rest of the replication cycle doesn't follow as well. Unfortunately, the flu virus mutates so rapidly that amantadine is probably no longer useful. And if it is, it has to be used very early on in the infectious cycle. So if you, sort of like if you feel like you have been exposed and you worry about developing symptoms, then you start taking amantadine, or as soon as you start feeling symptoms, you start taking amantadine. And it doesn't prevent you from getting flu infection. It slows down the progression, and it reduces the duration so that you don't have flu for that many days. But anyway, so this is the process of attachment and uncoating. And then what happens after uncoating? So we're again at the same step: attachment, uncoating occurred, and now we'll start looking at what happens to the viral RNA genome beyond that step. So viral RNA will then have to be carried over to the nucleus, because for human cells, any transcription occurs within the nucleus, right? Transcription doesn't occur within the cytosol for us. Although the virus came with its own RNA-dependent RNA polymerase, right? But any other machinery that is required to sort of make that the copying process easier, it's going to occur within the nucleus for us. So the RNA genome has to enter the nucleus, and within the nucleus, you are going to have the RNA-dependent RNA polymerase of the virus convert the negative strand RNA into positive strand, right? And one of the things that positive strand RNA is going to be used for as a template by the same RNA-dependent RNA polymerase to make lots of negative strand RNA genome. Right, so you're going to have this one purpose for that positive strand RNA. And the other purpose is that positive strand RNA would be synthesized for each of the segments, so it can be translated by our ribosomes, okay? And so each one of these RNA segments are being converted into positive strand for two purposes. One, so that they can serve as a template for further synthesis of the negative strand RNA genome. And two, so that we could use those positive strand RNA as mRNA to translate the corresponding protein. Now, one of the things that is a problem with within a host cell like ourselves, a eukaryotic cell, is that our RNA is processed as this 5' cap, and that 5' cap is required by our ribosomes to recognize this as a self RNA, so that it will only translate it if it recognizes that. So one of the things that the flu virus does is that it snatches, it's referred to as cap snatching. It takes about 10 base pairs and the 5' methylated cap region of our RNA and uses it to prime synthesis of the positive strand RNA. All right, so only for the one that will be used for translation purpose, not for the one that will be used for genomic purpose. Okay, so you're going to have cap snatching happen just to allow this now viral positive strand RNA to be now translated by our ribosomes, right? So depending upon the where the viral proteins are needed, right, translation would occur on either bound ribosomes or free ribosomes. So free ribosomes are found within the cytosol, and they're going to synthesize, proteins starting with RNA segments that code for matrix proteins or that code for some of the accessory proteins like the nuclear export proteins, NS1 and NS2, or nucleocapsid proteins, right? So these are all proteins that need to be soluble because they need to move back into the nucleus so that they could help export or help the RNA viral genome leave the nucleus, okay? Or it could just be the nucleocapsid proteins that will sort of coat the newly synthesized negative strand RNA. So it looks like, all of this pink layer is that NP protein that has to move back into the nucleus. So there are some proteins that would be synthesized in the cytosol for that region because they need to be soluble, right? Because they take part in assembly within the nucleus or within the cytosol. Then there are other proteins such as spike proteins, NA protein, HA protein, M2 protein. These are all going to be synthesized via bound ribosomes because they need to be inserted into the membrane. And you'd remember that for our cells or any eukaryotic cell, any protein that needs to be inserted into the membrane's journey starts with the rough ER. So you're going to have bound ribosomes synthesize these proteins, and as they synthesize them, they will also insert them into the membrane. And then from there, proteins will move from rough ER into Golgi and from Golgi into the membrane via vesicular pathway. Right, so vesicles are going to take, like a little vesicle is going to bud off from the rough ER, and it's going to have these spike proteins in it, and it's going to fuse with Golgi. So they'll move to Golgi, and from Golgi, new vesicles will form like secretory vesicles, and they'll move to the surface so that eventually these spike proteins are going to make their way up to the surface of the host cell. And so you're going to have all of these spike proteins present at the surface of the host cell. The M matrix protein is going to basically bind to the inner surface of your plasma membrane, or just kind of form a layer there. And then you're going to have these 8 segments. Okay, you can't just have 2 of segment 1 and then the other 6 segments, right? You have to have one of each of those 8 segments come together and form a new virion, which is a very complex process, right? And there is evidence that, this is controlled by actually, certain machinery. But you can imagine that there will be some mistakes made where there will be viruses that might not carry each of the 8 segments, okay? So a lot of flu virions that are synthesized can be defective in that they don't carry the entire genome. They carry 8 segments, but they don't comprise the entire genome. They might have 2 of something or 3 of something, okay, or 2 of 2 different things, and so on. In order for a virion to be, infective and go on and carry on the life cycle, it has to have each of those 8 segments, which can get tricky as the virions are being synthesized and as they're leaving the host cell via budding. And one of the things that has to happen is, of course, this is a complex process again, which I don't know if we know enough about, but one of the things that has to happen is that as the virus is leaving the cell, remember, it still has the HA spikes on it, and the host cell still has the protease on it. And so you can have the HA spikes begin to interact with the protease and never be able to leave, okay, unless the unless the neuraminidase was around. So neuraminidase job is to, or NA spike, its job is to cleave off that final interaction between HA and the protease as it's leaving. So sometimes we refer to it as NA helps to cleave the mucus as the host, as the virus leaves the host cell. But so if you don't have the neuraminidase around, then the virus will be unable to leave the cell, which is kind of form a bud, but would remain at the surface. So that is where the Tamiflu was acting, that it was preventing the exit of the virus. If the virus doesn't exit, eventually this cell that is infected will be recognized by your immune system and it will be destroyed. But as long as you don't synthesize new virions, they don't go on and infect neighboring cells and don't prolong the infectious cycle, right? So Tamiflu worked in that way. And of course, once you've synthesized new virions, they can go on and infect, subsequent host cells. Questions on this? We're good? Okay, guys. All right, so one of the things that we hear a lot about is, booster shots or yearly flu shots. And, why do we need to take these flu shots? We need to take flu shots because the virus mutates very, very rapidly. And it doesn't mutate as rapidly as the COVID virus does. It mutates about 300 times the rate of other microbes. And it amounts to about 2 times 10 to the minus 6 to 2 times 10 to the minus 4 mutations per site per progeny or per round of replication. So it's higher than bacteria, say, but it's not as high as COVID, which can have 1 times 10 to the negative 3. All right, a time, I think 1.12 times 10 to the negative 3, which is several times higher, at least 100 times higher than that of the flu virus. Okay, and so where does that rate of mutation come from? So the rate of the mutation comes from the RNA-dependent RNA polymerase copying RNA into RNA. When we talk about DNA dependent RNA replication, which is what happens in our cells, right? We talk about DNA dependent RNA production. That involves DNA polymerases, and DNA polymerases have a proofreading activity. RNA-dependent RNA polymerases do not have the proofreading activity. So if they insert a wrong base by mistake, that base is going to be retained. And any proofreading mechanisms that the host cell has are at the level of DNA, not at the level of RNA. All right. So and it's those mutations are going to be retained. And so there are 2 types of changes that occur in the flu virus. One we refer to as the antigenic drift, which is a slow process, and it owes its foundation is within that lack of proofreading activity within the RNA-dependent RNA polymerase. And so mutations will be made as HA and NA genes are copied. And because HA is what's involved in binding to the receptor, right? Mutations within HA are of a higher importance, right? So in antigenic drift, what you're seeing here is the HA trimer. And there is the region that binds the host cell sialic acid receptors. And the blue region represents the part of this trimer that binds specifically to the receptor. And there's a green region. These are sites that we refer to as antigenic sites because these are recognized by our immune system, specifically our specific immune system and antibodies produced by B cells, okay? So when we're talking about this antigenic drift, anytime you have mutations that occur in the blue region and they are deleterious, then they prevent the ability of this HA from binding to the host cell receptor. And so they're beneficial as far as the host cell is considered, because the virus will be unable to bind to the receptor and no infection would occur. So from viruses perspective, that's a bad thing, right? So they want to avoid mutations in that site. Do they specifically avoid it? Probably not. Mutations do arise and within that blue region as well. But because those viruses then are no longer infective, we don't worry about them, right? We worry about the ones that keep their binding site intact, but change their antibody binding site. So every time we get vaccinated, we have a immunity memory that is being induced that learns to recognize, this green site. And every time a virus mutates and changes these green antibody binding sites, then our immunity memory doesn't work. The antibodies that we have going around within our body trying to destroy this virus no longer work. And so we need a whole new, vaccine that helps to protect us against this newly mutated virus. Okay, so this is going to result in new vaccines forming every year, right? So generally, you would have organizations like the World Health Organization or CDC that are going to continue to monitor the types of flu viruses are going on within a population, and they will use those viruses to synthesize vaccines. But every now and then, right, there comes what we refer to as an antigenic shift, where the virus changes dramatically. So it's no longer just changing a little bit. It's not changing, small mutations that are changing the antibody binding site. It's rather a brand new HA in combination with other spike proteins that results in formation of a potent strain that none of the vaccines that we have in use are effective against. So how do those types of strains arise, that sudden abrupt change in antigen where a whole new strain of virus is generated and our immune system isn't ready to combat it? Our vaccines don't work against it. Okay, so we need to synthesize a whole new vaccine, and to synthesize a vaccine, it could take up to 6 months. The flu vaccine that we may take sometimes in September, synthesis might begin sometimes in January, right? So, you can have brand new strains arise during that time, right, between January and September, and or after September, and so the vaccination process, its synthesis can take a while, even if you can isolate that new virus. But in this case, what we're seeing here is each one of these parallel strands, white strands that you're seeing in a background, are representing the 8 segments of individual viruses. So there are human viruses at the bottom of this chart. There is the bird avian flu virus at the top. Then you have the classical swine flu virus fragments. And then you have triple assortment swine, and then we have H1N1 outbreak that we saw, in 2019. And you can either focus on the triple assortment swine or you can focus on H1N1. But the idea here is, let's focus on H1N1. The idea here is that this virus resulted from infection of a swine that was infected with swine avian flu virus as well as the human flu virus. So it gave rise to a brand new combination where you have, say, one swine that is infected by both of the avian flu as well as the human flu kind. And it has to be a single cell that is infected by that same, by those 2 viruses. And so that single cell has 16 strands of RNA segments. And so those RNA segments can be reassorted. So you can have RNA viruses that are completely, that are being synthesized that might be completely just the flu virus, meaning they have all 8 strands of the avian flu virus, or they have all 8 strands of the human flu virus, or they can recombine them and have some that are from the avian flu and some that are from the human flu. So it results in a brand new virus that has um, recombined those 8 segments between the avian flu and the human flu virus. And now you have a brand new virus, which has this brand new combination of H and N genes. Okay, so that's essentially what's showing you that with the H1N1 virus, H1 was derived from the classical swine flu virus, meaning it infected mainly swine. And then N was derived from, avian-like swine flu virus. So you can basically trace back each of these fragments to individual, fragments all the way back to either avian flu or swine flu or human flu. So I want you to sit down. I'm not going to ask you questions specifically on this, but hopefully you understand how just infection of the same cell by different flu viruses can result in reassortment of those RNA genomic fragments into brand new combinations. And that is what's behind the antigenic shift. And that is what causes, these outbreaks or pandemics, something like the H1N1. And these are the ones that vaccines will no longer be effective against, because the vaccines were generated using the classical, say, flu viruses that were going around. So when we hear words like H1N1 or H2N3, what does that mean? So it refers to the number of different alleles for each of the H and N spikes there are. For H spikes, there are, I believe, 16 genes. And when you look at them, H1, H2, and H3, they classically infect humans, but H1 also infects human, swine, and birds. Okay, and then you have from H4 all the way to H15. These are all either the avian flu viruses or some other mammals, such as farm animals. So you have different flu viruses infect different host cells because of the type of spike they have on them. So human viruses will have always H1, 2, or 3 as part of their name. And then you have the Ns, right? So you can have N1 or N2 and N3. And similar to what we talked about with the HA spikes, the NA spikes can have multiple alleles as well. And, they are present within viruses that are specific to certain host cells. So N1 and sorry, what am I doing? I'm doing something. Okay. All right. So you can have N1 and N2 containing flu viruses that can infect both humans as well as the swine, as well as the birds. All right. So the combinations of the name, the point that I'm trying to get to, is dependent on the specific allele that this virus in particular has. And recombining these alleles and having newer combinations can result in antigenic shift, which is basically a brand new combination of the virus that our immune system may not be able to recognize and destroy as easily. Now, when it comes to formation of the vaccines, vaccine essentially is just taking a weakened virus or just part of a virus and injecting it into our body and allowing our immune system to recognize that and mount a specific immune response, which results in activation of B cells and T cells. And so, you have the memory of this encounter remain as some of the memory B cells and T cells. So if you have a natural encounter to this virus, down the road, those memory cells would be able to mount a much faster immune response. So you might not develop any symptoms at all, or you might develop symptoms that are very, very mild. So this is basically priming your immune system to just be prepared so that it's able to recognize and destroy the virus and virally infected cells. So the flu vaccine is produced in different ways. You have the egg-based vaccine, where because birds have a receptor for flu viruses, you can inject the flu virus into bird eggs, such as chicken eggs, and you allow the virus to be replicated within that chicken egg. It's a complete system with its own nutrition, a way to get rid of carbon dioxide and acquire oxygen. And so it's a sustainable system for growth of this virus. And so every year, millions of eggs are used to grow flu virus. And you might have heard, like, are you allergic to an egg question when you're trying to get a vaccine? It's basically referring to the fact that this virus was grown within eggs. And, after they've been grown, of course, they're isolated and purified from those chicken eggs. And, that is what we get injected with. You can also have cell-based vaccine, where the same process of growth can occur within cells of mammalian origin in a petri dish. But of course, at a industrial scale, you're going to have bioreactors that have, lots and lots of cells that are growing at once, acting as host cells to grow these viruses. And then you can either take those viruses, weaken them, or just break them down and then use just the part that is antigenic for injection, or the recombinant vaccines, which don't have any virus at all. With the cell-based vaccine, or especially egg-based vaccine, you can have weakened virus, which means that it is either intact and completely inactive, so that it is not able to cause any type of infection, or it could be a virus that is able to cause an infection at a very, very small scale, so that it can result in amplification of the number of viruses within the body. And, of course, they are targeted. And then finally, recombinant vaccine, where instead of injecting people with the whole virus, and especially a weakened virus, which still retains the potential of replication, why not just take the gene that encodes a particular spike, like the HA spike or the NA spike, or both, and then either inject people directly with that gene, right, either in RNA form or the DNA form, and then allow our cells to transcribe and translate the corresponding protein, insert it in its membrane, and allow our immune system to recognize that. But of course, it spares us from having to worry about a full-blown flu infection from this particular method. So you can also just sort of harvest those individual genes, and then use those genes to synthesize proteins in vitro, and then inject those proteins, or allow our own cells to synthesize those proteins, completely bypassing the in vitro synthesis of proteins altogether. But the end result is the same for all of us, that our immune system would be primed to interact with the flu virus. Any questions on that? Okay, so I think I just have enough time to talk about the COVID virus, and then if I feel like we have time, I'll go over HIV. If not, then this will be the last one we'll talk about. So SARS-CoV, you guys know a lot about it. SARS stands for the severe acute respiratory coronavirus, and it's very, very mutagenic. And so people have been studying it for the longest time. It's not a virus that was just discovered. We've known about this for centuries because it causes common cold. But just like the antigenic shift you saw in flu virus, where you have the sudden rise of mutagenic strains or mutated strains, we can have that similar phenomenon occur within the SARS-CoV virus as well, or COVID virus. And so as I mentioned, its mutational rate is 10 to 100 times, or 100 to 1,000 times rather, higher than that of what you would see in flu virus. And it's similar to the flu virus, right? It has the same envelope. It has all of those spikes, and those spikes give it the appearance of a crown. So therefore, we refer to it as a coronavirus. And it also has an RNA genome. And the RNA genome is quite large. Okay, the 30 kb large. It's one of the viruses that have the most complex genome. And there are several sort of different strains of this virus around. And when we compare their genomes, what we find is some structural main protein encoding genes, and then there are others that we'll refer to as accessory genes. And accessory genes are generally involved in pathogenicity, meaning going into the cell, controlling the cell's machinery, and helping to basically cause disease. Okay, and so for the purposes of the virus itself, the four main structural proteins is going to be the spike protein, the envelope glycoprotein, the membrane glycoprotein, and the nucleocapsid protein. So those are the structural protein. And then there's also the 5' end of the genome, this largest portion, about 20 kb, that encodes a replicase. Okay, and so the replicase is going to code for RNA-dependent RNA polymerase. All right, so 20 kbs, 2/3 of its genome is dedicated to synthesizing an enzyme that will help the host cell copy its RNA genome into RNA. Okay, and so it's extremely important. And of course, if you have a lot of mutations occur within the replicase region and it abolishes the function of the replicase, and the virus can't, of course, continue its infectious cycle. So oftentimes when we're worried about mutations that are gain of function mutations for the virus so that it's able to infect more people, we're often talking about, obviously, the spike proteins that are a problem, right? Spike proteins that will begin to, that begin to have a higher affinity for the human receptor protein, right? So the genome is structured in that 5' end has the replicase gene, and then the rest of the 10 kb is zoomed in at the bottom here, so you're seeing it, and it's the ones that are important and that they're part of the structured protein is the spike protein, right? The M protein, and the sorry, is that the E protein? Yeah, that is the E protein, and then the N protein, which is the nucleocapsid protein, things that I mentioned already, right? So we have one strain here represented as this, then you have another couple of strains that are representing that remaining 10 kb region. And notice there's variation from virus to virus. So MERS-CoV was the Middle Eastern Respiratory Syndrome virus that was causing infections around, I believe, 2005, 2006, around that time. But we were able to, take care of it quite rapidly. But in any event, so this is what its genome looks like. Mainly 2/3 of it is the replicase, and the rest of the genes are sort of found in overlapping fashion within the rest of the 10 kb region. And of those, the S, E, M, and N regions are extremely important, and mutations that are in the spike region are going to be the ones that are responsible for, generating mutants that can much more easily spread. Question. 20 kb is 20 kilobase pairs. So I mentioned that the genome is 30 kilobase pairs, okay, 30,000 base pairs long. And 20,000 base pairs out of that 30,000 base pair region is just to code the replicase. Okay, so what happens when it infects our cells? So the SARS, the COVID virus that we have right now, it binds to these receptors that we call ACE2 receptors. These are found on our cells, and they bind a hormone called angiotensinogen 2. Its journey begins with the kidney, and you have it begins as angiotensin angiotensinogen, and then it, as it's passing through our lungs, it's converted by an enzyme into angiotensinogen 2, and then it binds to the ACE2 receptors that are found on our lung tissue, and it helps to control blood pressure, okay? It helps to lower the blood pressure. And so you couldn't possibly just say, well, why don't we make a drug against the ACE2 receptor and prevent its binding to these receptors? Because if we inhibit these receptors, we also abolish, partially control of blood pressure within our bodies. Okay, so it might work for somebody that has low blood pressure, but it's not going to work for most normal individuals, and it's going to wreak havoc with that entire regulatory mechanism that we have going on for the blood pressure regulation. But once it gets inside the cell via receptor-mediated endocytosis, it's going to undergo uncoating in a mechanism similar to what we saw within flu virus. And the RNA genome is not like the flu RNA genome in that, one, it's a positive sense RNA genome, right? And two, there's a single segment, so you don't have 8 segments of it to be copied. Now, because it's a positive sense RNA genome, it's ready to be translated. So you're going to have some pre-ribosomes begin to translate one of the genes, specifically the one at the 5' end. And you'd remember at the 5' end, you have the replicase. So the replicase is the first one that will be copied, and it will, the protein will be synthesized. It will undergo proteolysis to produce the various individual protein segments of that RNA-dependent RNA polymerase. And so that replicate transcriptase is essentially that RNA-dependent RNA polymerase that's being synthesized, okay? So that now it can use the positive sense strand RNA to convert it into negative sense RNA so that it could be copied back into positive sense RNA, right, using the same RNA-dependent RNA polymerase. And it will also copy the original genome into positive sense RNA so that individual genes corresponding to individual proteins will be transcribed. And when you look at these RNAs, we refer to these RNA as nested RNA, all right? And what that means is that they can fit into, they overlap with one another. They fit into, the sequence overlaps. And so, it's kind of like if you've ever bought like Tupperware that you have, from the largest to the smallest, and they all fit into one another. That's called like a nested Tupperware. So it's kind of like that. You have these nested RNA that are being synthesized, where the 3' end is similar to them, but the 5' end is different. And that makes sense, right? 5', the first one should have, say the first nested RNA is going to be copied so that the entire thing is there. And so this protein would be synthesized using that particular RNA. Then the second RNA might be missing this part, the 5' end part, and then only the S gene is and beyond is being transcribed, and then 3A and beyond is being transcribed, or E and beyond is being transcribed. But the end result is that all of them share the 3' end. They differ at the 5' end. And then depending upon where these proteins need to be, whether soluble in the cytosol or embedded into the membrane, that is where the translation for these nested RNA would occur. And so for the spikes, of course, the translation of the corresponding RNA would occur on the bound ribosomes. And so the spikes would be inserted into the ER. And from ER, these spikes will be moved into the Golgi. And then notice that the assembly of this virus is occurring essentially at the Golgi complex. So this is where the envelope that this virus is acquiring, it's getting from. So it's getting it from the Golgi complex, and that is where sort of it's budding off of, okay? And so that's where the assembly is occurring. This is where the nucleocapsid and any proteins that need to be, included into the core would interact with the trans-Golgi complex, and you would have, the full assembly of the virus, and then eventually virus would leave the cell after maturation and is free to go on and infect further cells. Now, I didn't mention where a lot of the mutations are occurring. So the concept is the same as we saw in the flu virus, that you have the RNA-dependent RNA virus that is using RNA to make more RNA. And this RNA-dependent RNA polymerase doesn't have the proofreading activity. You have a large genome, 30 kilobase pairs, that this RNA-dependent RNA polymerase has to copy. So it's going to make lots of mistakes, okay? And those mistakes are not going to be, corrected. But in terms of those gain of function mutations, generally, from the current pandemic, the data suggests that the rate of mutations is about 2 mutations per genome per month. Okay, so it's quite high. So that's the basis for it, that it's coming from the fact that the RNA-dependent RNA polymerase lacks the proofreading activity, and it also has to copy this large genome. But why retain those mutations? Any mutation that allows the virus to have a greater fitness, which means that it is able to infect the host cell better and also avoid the immune system better, it will be retained. Okay, and so these mutations arise spontaneously, right? These mutations arise if you were to grow this virus in a petri dish. It doesn't have to be growing within the human host cells. It's because it's due to that inherent lack of proofreading in the RNA-dependent RNA polymerase. But they are selected for within the host cell because some mutations allow the virus to infect the host cell with much better ability than others, okay? And then there will be other viruses that will not only have enhanced binding ability, but also less pathogenicity, less, sort of antigenic nature, so that it doesn't attract the immune system as much, okay? So over time, as the viruses mutate, they tend to become more tamed, okay? The greater the number of mutations, the lesser the likelihood that they will all be gain of function mutations. So as this virus is mutating, it's not necessarily a bad thing. So we're really worried about this Omicron virus. It has about 50 mutations, a lot of them in within the spike region. There's a possibility that some of those could be gain of function mutations that it will result in the virus becoming more infectious. But the greater likelihood is that the virus is actually going to become, less infectious, or at least have this greater, fitness where it is able to infect our cells, but also avoid the immune system, so that it's able to continue to infect, produce more of its kind, cause mild symptoms called mild disease, and doesn't result in as many severe disease instances and hospitalization cases. So when we talk about the vaccines for this particular virus, you guys have all taken the vaccine. So there are RNA vaccines. So RNA vaccines have to be injected as RNA, but RNA has to be picked up by cells. So generally RNA is coated by some sort of a lipid membrane. And so that lipid membrane fuses with our cells. And when the viral spike RNA is delivered within our cells, it will be translated by bound ribosomes, and it will make its way up to our membranes, and that's where your B cells and T cells would, learn to recognize it, and you would have an immunity form. But because you don't have the weakened virus or inert virus that is being injected, you don't have that possibility of that virus regaining its strength and causing the disease itself. There's also the Novavax, which a lot of people are waiting for. and especially with the current arrival of the Delta variant and the Omicron variant, which is protein-based, which is essentially just the spike protein. So it's not just the RNA, but rather the spike protein. There's others that have the weakened virus as well. Okay, we're not using them. Of course, we're here. We just have the Moderna, Pfizer, and Johnson and Johnson. Johnson and Johnson is delivering the same RNA via viral vector. So this vector has spikes on it that allows it to enter our cells, versus where RNA was being coded by lipids, and lipids were automatically able to fuse with any of our cells and result in synthesis of the spike protein. With the viral vector, the vector must have receptors on some cells, and those cells are the only ones that would be, of course, infected. How much time do we have? I think I'm going to stop. Our time is up. Anybody has any questions regarding this, please let me know.
Professor?
Yes.
So you didn't cover the HIV. Are we going to have that in our exam? Or
Recording stopped.
No. I've talked about it a little bit when we talked about the drugs that target the viral replication. So I wasn't worried about HIV too much, and that's why I left it at the back. And I think I also discussed it a little bit when we talked about the viruses themselves. I talked about it as a retrovirus. So I think I'm gonna just stop now, and I'm just gonna have coronavirus, flu virus, and the lambda phage. And for the final exam, you don't have to worry about HIV.
Okay, great. Thank you.
Okay. Any other questions? We're good. All right, guys, good luck on the final exam. I hope that you were able to learn something and take with you, enough from this course that helps you understand, that microbes aren't all bad. And even with the bad microbes, we can certainly study them and learn more about them and devise ways of, controlling their growth, whether outside of the human body or within the human body. Good luck on your lab final this week. And as I've mentioned, I posted the date for your lecture final on the Blackboard, and the final will cover the last 4 lectures. As I mentioned earlier, I'm going to leave these recordings on Blackboard until the day of the exam, and then I will remove them before the exam. Okay, good luck. And if you guys have any questions, if you need to see me briefly, please email me and I'll be more than happy to accommodate you. Okay.
Professor?
Yes.
Do you mind if we can make the exam like in parts?
I think I will. I think I will make it so that it gives you time to kind of stop in between and take it in pieces. That's completely fine by me.
Thank you so much.
All right. Take care, guys. Thank you, everyone. Good luck.